So fusion proteins, these are often considered to be the most undruggable of undruggable targets. And it turns out that they're often really precise, like ultra-precise targets of interest for pediatric cancers that are very rare, including cancers like pediatric rhabdomyosarcoma, which only impact, say, 500 patients per year. And so as I started to think about what's the most undruggable tough problem target I can think about, and wow, CDK9 and MYC seems to have an impact, it would be fusion oncoproteins. And during my time at MIT, the NCI, through the Cancer Moonshot mechanism, created a consortium of different investigators with different backgrounds to try to make an academic impact on these different cancers. So if you're not familiar with what a fusion protein is, here's an example. PAX3 is a transcription factor, and this is what it looks like. FOXO1 is a transcription factor. This is what it looks like. And when a kid is unlucky, these two different proteins come together and fuse and form a fusion that will drive cancer. The other parts are not oncogenic. This is oncogenic. And it's very disordered, very spaghetti-like, so no one has been able to really come up with drugs for these sorts of targets. So we came back to our SMM platform. A number of colleagues here at MIT ran screens, and we identified a bunch of hits, and we've been prosecuting these hits in different ways. I'll just make reference to an ongoing IP story in our lab now. We found a binder to one of those fusions, the PAX3-FOXO fusion. We were able to demonstrate binding and target engagement in cells. And then we converted the compound into what we call a PROTAC. So this is a silent binder. It doesn't impact the function of the fusion, but we could deliver this target now to the proteasome to be degraded. And so this is now, we have filed on this. This is now the starting point for a much larger project in a direction in our lab, something I'm excited about. And to sort of stir the pot, I'll say, I know what to do with a CDK9 inhibitor from a translational perspective, but when you have a drug that could impact, say, 500 patients a year, I can tell you that all of my friends in pharma will not advance that. So we need to think about other types of partnerships, maybe with the government, with different foundations, Summers Way and Emerson Collective. Emerson Collective actually funded us to get some of the preliminary data. And coming back to my PhD advisor in this new institute, maybe we work with some kind of other team to try to bring our probe all the way to a drug for these patients.